Hi, this is Miss Litton, and this is a very busy lunch room, and we are doing an AP review on Big Idea 2, how biological systems utilize energy and molecular building blocks to grow, reproduce, and maintain homeostasis. And where we are on the review is we're under Enduring Understanding 2B, and so let's jump down to 2B, or not. That was kind of funny, come on. So we're on 2B, and we are on part like halfway through, which is on slide 43. Where are we? Slide 43. Oh, no, we're just on 2B. Never mind. Never mind. Okay. So growth, reproduction, and homeostasis require that cells create and maintain internal environments that are different from external environments. One word to explain all of that is, what's the key word in that whole statement? Homeostasis, exactly. Okay, so the first thing we want to talk about is you create a barrier that keeps the inside different from the outside. And so we're talking about the cell theory. And so if you remember the three parts of this cell theory, all living things are made of cells, and that cells are the structural and functional units of organisms. That's how they work. And that cells can only come from pre-existing cells. And if you remember, we also discussed in order. Remember, you guys, I'm doing a review right now, so you need to nod. If you want to have a cell work, if you remember, cells must be very, very, very what? Small, so that you have a large surface area per volume, because all the interactions are going to be across that cell membrane. We then had a very long discussion about the protein functions on that cell membrane, on that barrier. But before I go any further, we talked about it being a phospholipid bilayer, right? Phospholipids with proteins interjected. And who can't freely cross this cell membrane if you are what? Large or charged. Good. And so we looked at things like non-charged molecules, like fats can dissolve into fats and then they can cross. Large molecules can't fit through. Water wouldn't generally go through because it's a polar molecule, but it does move because it's so small. And additionally, there are protein channels called what? Aquaporins that allow it to pass. Good. Okay, so then we talked about ways that things can cross the cell membrane, and we talked about passive and active transport. If you hear the word active transport in it, it's going to require what? Energy. Active transport requires energy. Okay, so you want to make sure you remember that. Now, passive transport, you have some options on passive. You have straight up diffusion, where you're going through the phospholipid bilayer. But then if you need to use a protein to do your diffusion from a higher concentration to a lower concentration, then it's facilitated diffusion. You're using a protein. Okay, check with the person next to you and see if are they okay with everything so far. Got it? Okay, there are other forms of active transport besides the sodium-potassium pump. Can anybody name what those other forms of active transport are? Exo and endocytosis, perfect. Okay, so then let's look at all the different ways you can cross. So you see straight up diffusion going from higher to lower. You must not be large or charged if you're able to do that. You can use a carrier. Generally, when you see a carrier, you're interacting with it. A lot of times those are going to cost you energy. You might be going against the concentration, but maybe not. A channel, also very specific. And then osmosis is specifically the diffusion of water across the semi-permeable membrane. And you can see here, this must be a what? You can go through the phospholipid, but here, what must this thing be right here? What would you call it if this is water? Aquaporin. Good. Okay, so this, again, another summary. The only thing that's kind of added on here is that this is showing you phagocytosis, which is cell eating. What is cell drinking called? Penocytosis. And phagocytosis and penocytosis are both examples of endocytosis. And going the other direction is exocytosis. Perfect. Okay? This is just a review of a carrier. and a channel. We talked about cystic fibrosis. Sodium potassium pump. At this time, we learned this the first time, the sodium potassium pump, that was different. Now, when you think sodium potassium pump, what probably comes to mind? Neurons, right? And what does the sodium potassium pump help set up? Are you saying the resting potential? Okay, the resting potential, right? And the resting potential for a membrane is what? Sorry, taking a bite. Minus 70 millivolts. And that's because you have more what on the outside? Sodium on the outside than you do potassium on the inside, even though they're both positive charges. Why else? Negatively charged ions on the inside. And what's the third thing? Leaky potassium gates. Good. So now we understand about the sodium potassium pump. Okay, cell recognition. This should have a whole new meaning now that you have studied what? Immune system, right? Who's going to be using these primarily? What type of cell? Yes, a type of white blood cell. Be more specific. T cells, right? We talked about MHC1s and MHC2s. And just as a review, if you are holding an antigen in an MHC1, you will be done. You're going to get killed. Who's probably going to kill you? A cytotoxic T cell. Excellent. Okay, or who else could kill you? Natural killer cell, right? And if you display an antigen on an MHC2, you'll get help, right? You're usually an antigen presenting cell saying, I've killed this thing and I'm presenting part of this antigen to you so you can help me fight this infection. All right, and then receptor proteins. Where you can think about this, you can think about hormones. What kind of hormone requires a receptor protein? What? You could really tell me all hormones. It's just where the receptor is located. If the receptor is located in the cell membrane, on the outside, it's got to be a peptide or protein hormone, and it triggers a second messenger inside, right? And if it's a steroid hormone, the receptor will be inside the cell. And usually steroid hormones impact what? Transcription, translation of proteins, and they work what? Slower or faster? Perfect. They said slower. Okay, and then an enzymatic one, you could be thinking about the example that I gave you. Do you remember with the second messenger? Do you remember this one? All right. Aquaporins we already did. If you're not a peripheral protein on the edge, what do you call the other one? Starts with an I. Integral, good. Okay, we'll skip that. Okay, osmosis. Bye, sweethearts. Have great breaks. Make good choices. Rest. Osmosis, three things. Diffusion of water across a semi-permeable membrane. Let's describe some things. How would we describe this side right here? Hypertonic.
Thank you, son.
You're welcome. And does it have more or less OASs? A hypertonic side. Do you remember what OASs are? Osmotically active substances? Did I teach you that? Oh, okay. Yeah, I taught you something new. Osmotically active substances. So these are things that are going to interact to affect the flow of water. Okay? So this side is hypertonic. It has more OASs, osmotically active substances. Because if I put rocks in here, is it going to have the same effect as sugar or salt molecules? No. Sugar or salt molecules are going to interact with water, and that water is going to be bound water, and that's going to influence the way water flows. Okay, so this side has more OASs. It's hypertonic. Does it have more or less osmotic potential? Less osmotic potential. Good. Okay, but it does have more osmotic what? Pressure for water to come in. And conversely, on this side, it's not hypertonic, it's hypo. It has less OASs. What else does it have? More osmotic potential. What else? Less osmotic pressure. Good. And we know water must flow from the hypo. It's still flowing both ways, but the net change in water is going to be from the hypotonic solution to the hypertonic solution. Muy bueno. Okay, and so then isotonic solution is same. Now, it could be same because it has the same osmotic pressure on either side, and that could be generated by a plant cell wall pushing back, even though it is a more hypertonic solution inside. Do you understand what I just said? Okay. And then hypotonic solution makes the cells swell. Hypertonic solution, crenation. All right, then we talk about water potential. Water potential is for plants. Water potential is a factor of two things. I guess that's it. Okay, I give up. Is a factor of two things: pressure potential and osmotic potential. Okay, pressure potential and osmotic potential. And so when, I don't know if I have this in here, let me just check. I do. Okay, so when you have a beaker of pure distilled water, right? Pressure potential 0, solute potential 0, it's open to the atmosphere, 0. But when you put this plant in here, if this plant has any molecules in here, if it's not distilled water inside, which it's not because it has all of the sugars and everything inside of that, then the solute potential is a negative number. Okay? Out here, your water potential is equal to 0. Inside here, your water potential, there's no pressure on this situation yet, is -2. So the best water potential you can ever get is 0. And then water will always flow from a higher water potential to a lower water potential. So water is going to flow in, and some water is going to flow out, but more is flowing in than out, and it's going to continue to flow in until the water potential on either side is 0. Now, how does that happen? Could it dilute the inside so much that it's not -2 anymore? Could it dilute it up to distilled water? No, it's always going to have stuff in there, right? But what's pushing back to bring it to 0? Cell wall. So when the cell wall pushes back the same amount that you're drawing fluid in, your water potential is 0, the pure distilled water around it is 0, so there's no more net change. Ask the person next to you if they understand that. Okay, so when we did that lab with potatoes, remember this part? And you had the different molarities. You remember this? Yeah. And you had mass here, and you had a line. Is this line right there the water potential? Thank you. It's the solute concentration. It is not the water potential. You use this right here to calculate the water potential, right? This is the molarity of the solution. And do you remember how we said the water potential is a factor of two things, right? The solute potential and the pressure potential, right? It's a factor of two things. Here, where there's no net change, the pressure potential is 0, so we can take that out. So now the water potential is just equal to the solute potential, which we know is equal to negative ICRT. Yes? I is the ionization constant. If it's salt, it would be Na and Cl, it would be 2. If it's sugar, it's just 1. You're welcome, sweetheart. Have a great break. So this is negative ionization. The C is this number right here. Okay, this molarity right here is the C. And you can use that to calculate the water potential. R is a constant. What is it? 0.0831 liters per bar per mole per Kelvin, I think. And then T is temperature in Kelvin. Okay, ask the person next to you if they understand that. Good now? Okay, here's facilitated transport. Facilitated transport, you're still using diffusion, going from a higher concentration to a lower concentration, but you're using a protein. We already talked about that. Here's active transport requiring ATP, and we already reviewed endo and exocytosis. This is receptor-mediated endocytosis. If you remember that, the molecules are binding to receptors on the proteins, and then when all the receptors are filled, then endocytosis occurs. And this is really a form of penocytosis. Okay, this brings us to the endosymbiotic hypothesis. What comes to mind when you think, what is it? And then what evidence do you have to support it? Think about that right now. What is it and what evidence do you have to support it? And tell it to the person next to you right now. What is it and what evidence do you have to support it? Okay, did they say how a eukaryotic cell could have evolved from a prokaryotic cell? Okay, and did they say that one prokaryote could have engulfed another prokaryote? Specifically, bacteria that were like mitochondria and like chloroplasts. And our evidence would be that mitochondria and chloroplasts have their own DNA. They have ribosomes that are about the same size as prokaryotic cells, that they are self-replicating, right? That they have two membranes, so they have one membrane that was their own and one as a result of endocytosis. And then you could have had membranes infolding in order to create the nuclear envelope and all of your vesicle-like organelles like lysosomes and ER. Questions on that part? Okay, then it comes to cellular structures. So you just want to remember what are the, what I would want to know is what are the key organelles and what role do they play in maintaining homeostasis? It's probably how I would function. So, for instance, mitochondria generate ATP through cellular respiration. I need energy in order to synthesize molecules, to transport molecules, so that helps me maintain homeostasis. You could talk about the nuclear envelope, separating the DNA from the rest of the cell in order to protect the genetic material that codes for the proteins. Okay, you could talk about ER involved in transport, right, of proteins to the cell. It helps protect them so they don't get digested because it puts a vesicle around them. Moves to the Golgi apparatus. It puts a mailing address on it in order to, so whether it's going to stay in the cell or get moved out of the cell. And I would just basically review each of the structures and what they do. The microtubule organizing center, which in animal cells contains centrioles, that helps send out the spindle fibers during mitosis to make sure that the chromosomes are distributed equally amongst the two daughter cells. Okay, and then obviously you would review your plant cell structures as well, along the same lines as what I'm talking to you about now. And know what they look like, but it's not so much that you're memorizing that is what they do, or maybe the series of things that happen. Like the proteins are synthesized on the ribosomes, and then the protein moves into the ER, and then it moves into the Golgi apparatus. And it could become a lysosome that helps in the digestion of nutrients, which would help maintain homeostasis, those types of things. And that pathway is here as well for you. And my friends, you just finished an enduring understanding, and we'll pick up with this one next time. Good job.
Where's the stapler?
Okay, hope you're having a good break and good reviewing. That was 18 minutes of fun.